Good evening to one and all in the second day of this national webinar. Within 5 minutes, we will start this webinar. The participants are joining. The room is getting full. Sending a messages. Hi everyone. Good evening to one and all. There are two resource persons today. The first resource त्याला ऍड करून घे तुला किधरून राहिला मला. Hello.
TM Bose, sir.
Yes, sir.
Continue.
Yes, sir. Thank you very much, Dr. Rakesh from the Department of Computer.
Kindly start video, sir.
Yes, sir. Within 5 minutes, we will start this second day of national webinar. There are two resource persons today, Dr. Naveen Congo, sir, and Dr. Farjin Paradia. The participants are coming in the room. They are joining right now. Within 5 minutes, we will start the second day of this national webinar. As this webinar is organized by Sri Shivaji Vidya Prasarak Sanstha's led Karma with Dr. P.R. Gogore Science College on the topic of potential application of indigenous bioresources. Our college is A+ grade college, and this is the second time we are organizing this national webinar in such a pandemic period of COVID-19. Very warm welcome to all participants. Within 5 minutes, more than 100 participants are right now joined with this national webinar. In a very short time, we will start this webinar. Just wait for 2 minutes more. Hello. Hello. फोन येतोय.
केम बोस है सर.
Thank you very much, sir. Dr. Rakesh. पुन्हा एकदा आपलं या ठिकाणी सर्वांचं स्वागत करतो. आज आपला नॅशनल वेबिनारचा दुसरा दिवस.
Yes sir.
श्री शिवाजी विद्याप्रसारक संस्थेचे कैलासवासी कर्मवीर डॉक्टर पारगोगरे विज्ञान महाविद्यालयातर्फे आयोजित हा नॅशनल सेमिनार on
सर
the
शायद
subject
अपने पास 1 GB तो भी चाहिए.
potential and application of indigenous bioresources. या
ठीक है
नॅशनल
सर, आप
वेबिनार
करो
साठी
तो सही.
आपल्याला
देखते हैं फिर, आप जल्दी ये कर लेना फिर फाइंड अप.
डॉक्टर मीनू पराबिया, डॉक्टर फाल्गुनी,
मतलब क्योंकि मैंने मतलब मेरा ऑब्जर्वेशन
Naveen Congo sir, and Dr. Faraj Parabia. So this is the second day. Yesterday there was a very fantastic lecture by Dr. Minu Parabia sir and Dr. Falguli Seth ma'am. I think so it is very fruitful for the Lyme Science family, and this is a good platform to exchange the ideas and share our knowledges with this webinar platform. Within a very short time, we will start the second day's resource person talk. Initially, Dr. Naveen Congo sir will enhance our knowledge. Thereafter, Dr. Farjin Parabia will enlighten us in the subject of these life sciences regarding the potential and application of indigenous bioresources. Here we can say that flowers will not grow if the stems doesn't allow it. Similarly, our management, our principal, directors, there is a support from this side, then and then only it is possible to organize such event in front of you. The participants are coming, they are entering into the Zoom classroom hall. More than 100 participants right now entered in this classroom. Within a very short span of time, remaining participant will arrive. We will start it after 1 minute. Stay connected with us.
Hello.
Hello, Naveen sir. Are you there?
Very good afternoon.
Hello.
Very good afternoon. This is Professor Naveen.
Okay, sir.
Are you able to see me?
1 minute, sir.
Okay.
You start your camera.
Okay. Yeah.
Hello.
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 Hello. Are you able to listen?
Yes. You are
Okay.
visible, sir.
Okay, great.
Okay, sir. Thank you, sir.
So can I start with my presentation?
No, sir. 1 minute, sir. KM Bose, sir. Please start the session.
Okay, sure. Take your time.
Okay, the resource person just arrived right now, so I apologize for a delay of 5 minutes. So, good afternoon to all the dignitaries, distinguished delegates, ladies and gentlemen. It gives me immense pleasure to extend to you a very warm welcome on behalf of Sri Shivaji Vidya Prasarak Sanstha's late Karmavir Dr. P.R. Gogre Science College, Dhule, who has clubbed all of us for this national webinar on the topic of potential and application of indigenous bioresources. I heartily welcome chief patron of this national webinar, Honorable Professor P.P. Patil sir, Vice Chancellor of Kavitri Bahnabai Chaudhary North Maharashtra University, Jalgaon, Honorable Baba Sahib Kunalji Patil, Member of Legislative Assembly of Dhule Rural and Chairman of Sri Shivaji Vidya Prasarak Sanstha, Dhule. Now, I would like to invite Dr. M.V. Patil sir, Principal of Sri Shivaji Vidya Prasarak Sanstha's late Karmavir Dr. P.R. Gogre Science College, Dhule, to give the preface of this national webinar. Dr. M.V. Patil sir.
Hello.
Yes, sir. You are audible.
Okay. Good afternoon. Namaskar to all my dear participants, dignitaries, eminent personalities, and dear participants. Today is the second day of the webinar. Yesterday we had two eminent personalities, Professor Parabia and Professor Falguni Seth, for this webinar. And today, Professor Congo from Sagar University and Professor Dr. Farzin from Veer Narmad University, Surat will be joining us for today's session. Friends, ours is a science college having single faculty, and we are having only science subjects, 9 UG courses and 13 PG courses. This college was established in 1956, and from that till today, we have achieved a great rankings in NAC as well as all UGC platforms. This is a reaccredited third cycle of NAC, and we have achieved A plus grade with CGPA 3.55. And we are also awarded with College with Potential Excellence and with the DST PIST program and by Star College by DBT. Uh, Our college is affiliated to North Maharashtra University, Jalgaon, which is now named as Kaviyatri Bainabai Chaudhary North Maharashtra University, Jalgaon. I am thankful to the guest of today's program, Professor Aulikar, Professor P.P. Patil, Vice Chancellor of this university, and Honorable Chairman of the Sanstha, Baba Sahib Kunalji Patil, for encouraging us to organize this program. Because without their support, and their guidance, it is unable to us. I am thankful on behalf of my colleagues. Today, Professor Congo will interact with us. So I hope that you all will enjoy and interact with him also. But due to the lack of time, in a interaction session, we cannot make more questions, or we cannot take more questions. That's why we are giving a mail ID or a numbers of the experts on the webinar group. So please, those who want to contact personally in details can contact them. I hope that the participants and the students who are admit or present today for this presentation will enjoy this session, and I congratulate you all for uh, being present for this webinar. Thank you very much. I hand it over to Dr. K.M. Borse for the further sessions. Thank you.
Thank you, sir. Thank you very much, Dr. M.V. Patil, sir, the principal of our college, already given the preface of this national webinar. Now there is a time for the floral welcome. Encouragement, enthusiasm, excellency is one of the major aspects of organization with an object of increasing the spirit of research and innovation. We feel proud to host this national webinar on potential and application of indigenous bioresources. Yesterday, already Dr. Professor P.P. Mahulikar, who is a Pro Vice Chancellor of Kavitri Banabai Chaudhary North Maharashtra University, Jalgaon, already inaugurated this webinar and became a chief guest of this 2 days national webinar. Once again, I would like to request all the dignitaries for lighting the lamp before the Ma Saraswati, who is a goddess of knowledge, music, art, wisdom, and learning. Light symbolizes brightness and prosperity. It removes the forces of Ma Saraswati, who is the goddess of knowledge, music, art, and wisdom and learning. हे सरस्वती माते, तुझीच राहो सदा मावर मंगलमय ममता. अजन आम्ही मुले माऊली, तुझी मागतो कृपा सावली. तुझ्याच पूजनी तुझीच गावो गुजगोरो गाता. हे सरस्वती माते, तुझीच राहो सदा मावर मंगलमय ममता. विद्यावृत विद्यार्थी जीवन घडो आचरण विशुद्ध पावण. आमच्यासाठी सदा वाहू दे ज्ञानाची सरिता. हे सरस्वती माते, तुझीच राहो सदा आम्हावर मंगलमय ममता. So after that, there is a presidential address. A Joel Baker quotes that vision without action is merely a dream. Action without a vision just passes the time, but the vision with action can change the world. So in the same manner, our Honorable Baba Sahib Kunalji Patil, the chairman of Sri Shivaji Vidya Prasar Sanstha and ML of Dhule Rural, have a visionary action, who always focus his energy and time on representing the peoples. So education is the most important vapor we can change the world. The person who have always consumed himself to change the society and the light of the darkness in the life of the students of Dhule Gramin area, the Panidar Amdar Baba Sahib Kunalji Patil, I request Principal of our college, Dr. M.V. Patil, to welcome Honorable Baba Sahib Kunal Patil by offering a bouquet. Principal Dr. M.V. Patil, sir, I request you to offer a bouquet. Please give a big hand. Thank you, Dr. Principal M.V. Patil, sir, who felicitates Honorable Baba Sahib Kunal Patil by offering a bouquet. Now, I would like to request Honorable Baba Sahib to address our audience of this national webinar on the potential and application of indigenous bioresources. Honorable Baba Sahib Kunalji Patil, who is himself called as a Panidar Amdar, his vision is the broad educationalist, and always inspire our team, our family. Honorable Baba Sahib Kunalji Patil, please.
Good afternoon, everybody. I welcome all the eminent personalities who are going to speak today on this webinar organized by our Science College. We are Sri Shivaji Vidya Prasarak Samstha and myself Kunal Patil. I am chairman of this 110-year-old Shivaji Vidya Prasarak Samstha. We are into science, arts, commerce, and we run engineering colleges, some medical colleges. This trust has an hospital of 800-bedded hospital. In all the fields, this Shivaji Vidya Prasarak Samstha is showing their presence. I'm really happy that this webinar has been organized by our Science College, and in this very difficult time of coronavirus pandemic, which has been affected the whole country, and this lockdown is affecting the day-to-day life of everybody, but still everybody is trying to work from home, and whatever possible we can give to the society, and whatever possible knowledge we can share, we are trying to do this. I'm really happy that everybody, all of you have joined this webinar, and I'm sure that all of you will share your ideas, your views, your technology, and you will definitely make this webinar very fruitful, and will share a lot of knowledge in this webinar. I really congratulate all of you to join into this webinar, and at the same time I'm not sure that how long this lockdown will continue, but I'm very sure that with the help of our doctors and our our trainers, and plus our police people, the whole society, we will definitely overcome this period of crisis, and with the great leadership, this country will definitely very soon will come back to the normal life. Till then, we should take care, and I again congratulate you and thank you so much for joining this webinar.
Thank you. Thank you very much. Honorable Baba Sahib Kunalji Patil inspires us. Thank you very much, Honorable Baba Sahib, for inspiring us. Now, I would like to admit my thoughts. A good event never ends in the world. They take only a pause and keep us awaiting for the next. Now, I call upon Dr. Harshal Bhamre sir, Head Department of Biotechnology, to introduce today's resource person, Dr. Naveen Congo sir, Professor and Head Department of Microbiology, Dr. Harisingh Gaur Vishwavidyalay, Sagar, Madhya Pradesh. Dr. Harshal Bhamre sir, please.
Good afternoon, honorable guest and all participants. I, Dr. Harshal Bhamre, Head Department of Biotechnology from SSBS, late Karmavir Dr. P.R. Gogre, Science College, Dhule, for the national webinar on potential and application of indigenous bioresources. Today, we are pleased to welcome Dr. Naveen Congo, a very special guest and a dynamic personality from Dr. Harisingh Gaur Vishwavidyalay, Sagar, Madhya Pradesh. Dr. Naveen Congo is a professor and head in the Department of Microbiology, Dr. Harisingh Gaur Vishwavidyalay, Central University, Sagar, Madhya Pradesh. He has more than 20 years teaching and research experience. Dr. Congo did his MSc with gold medal and doctorate in microbiology and biotechnology from Dr. Harisingh Gaur Vishwavidyalay, Sagar. He qualified NET as well as GATE and joined the same department in 1997. He has worked on xylenolytic enzyme from thermophilic fungi for his doctoral and postdoctoral work from Department of Biotechnology, Durban University of Technology, South Africa, and Department of Microbiology, University of Helsinki, Finland. He has more than 40 international peer-reviewed research papers, 12 books chapters, one textbook of microbiology, and one patent in their treasures. He presently research interests are production and application of microbial enzymes such as xylanase, mannanase, insulinase, L-asparginase, keratinase from fungal sources and generation of 2G bioethanol from agro waste using the fungal cellulose. So today it's a great pleasure to be having a great personality with us, and he is going to deliver his talk on microbial enzyme in generation of health promoting prebiotics. So please give your full attention and help me in welcoming him on the platform. Thank you, sir.
Thank you, sir, Dr. Harshal Bambre, sir. Now, I request Principal Dr. M.B. Patil, sir, to welcome Honorable Dr. Naveen Congo, sir, by offering a bouquet. Dr. Naveen Congo, who is a professor and head of the Department of Microbiology, Dr. Harishing Gaur Vishwavidyalay, Sagar MP, a renowned personality, now going to deliver a lecture in front of us. Give a big hand for the Dr. Naveen Congo, sir. He is going to thank you very much, sir, Dr. M.V. Patil, sir, for so feliciting Dr. Naveen Congo, sir. Now, I request Dr. Naveen Congo, sir, please enlighten us by your valuable guidance to our participants. We are waiting, we are eager to hear you, sir. Please, Dr. Naveen Congo, sir. Sir, unmute. Dr. Naveen Congo, sir, is there on the screen? Very shortly, we will audible him. Yes.
Yeah. First of all, a very good afternoon to all the delegates and all the students, all the dignitaries who are attending this webinar. And I profusely thank Professor Patil, Dr. Borse and his team and his vibrant team to make my afternoon a vibrant afternoon and a happening afternoon after so many days. Because during these tough times, when everybody is faced with lockdown and so many other things, so many constraints, it's good to talk about science, and it's good to talk about some real good serious science, and that to the title of this webinar is very apt. It talks about indigenous bioresources. So most of us are aware that our country has been bestowed with the bounty of nature, including Western Ghat, Himalayas, and beautiful rivers, beautiful forests. And among those lie the invisible, resource, which are called as microorganisms. So basically, I come from a microbiological background, and I would like to project to the young students that microbes are the cryptic and a very important bioresource, and they should be utilized to the fullest to solve the problems of health, environment, food, energy, and so on. So today I'm going to talk about one aspect of our work that I do with my 3-4 PhD students, and that aspect is about generation of prebiotics using fungal or microbial enzymes. So I would like to begin with my presentation, if I'm allowed to do so. I'm not able to share my screen. Why I'm not able to share the screen now? Dr. Borse, it looks like there is some problem with sharing of the screen. Can you listen me?
Sir, any problem with PPT?
Yeah, it's
Hello, sir.
kind of started some enterprise.
You send that PPT to me. I will run here.
Okay.
Hello, sir. You share that PPT,
Mm-hmm.
and I will run here.
Okay. I should have emailed it to you earlier also. But I mean, yesterday it worked well. Say new share, and here I go. It goes to some which is not required.
So you are not audible properly. Kindly send that presentation to our side. We'll run that.
Okay, I'm sending it by email.
Yes, sir.
Okay, here we go. Which mail should I use? Dr. Patil's mail? Should I send it to Dr. Patil's mail?
Yes, sir. Yes, sir.
Okay, in a moment.
KM Bose, sir. KM Bose, sir. You are on the screen. Kindly continue.
Rakesh, okay.
Yeah.
Thank you, sir. Within a 1 minute, we will start it because without the interference, there is no conference. This is
Yeah,
the
that is
new
true.
technology.
Last also you said this.
Yes, sir.
Why this is not?
No, you just take, sir, your own time.
Okay.
Be comfort, flash it out. Until then, I would like to say something here about my college. In my college, zoology, botany, and microbiology department, there is a Lions Club, Leo Club, Lotus Club. Lots of activities regarding these life sciences are going on here in the our college, SSUPS Science College. In the botany department, there is a Lotus Club. In the zoology department, there is a Lions Club. And in the biochemistry, life science department, biotechnology, microbiology, there is a Leo Club. So many activities we are conducting over here. In the same one of the activities here, national webinar on the subject of uh, potential of indigenous bioresources and their applications. Here we are going to present the fourth renowned resource person in front of you for this 2 days national webinar. On the same platform, we will in the future also, we will organize so that we can exchange our ideas among our family of botany, zoology, life science, microbiology, and biotechnology. We can share our ideas, our things, so that there will be the fruitful outcome of this national webinar. Similarly, everything. I think so. The problem is going to be sorting out within a fraction of second.
The file is there with.
Until up to then, I would like to announce one announcement here for the participants. Those who want to share their ideas here on the screen, just give their names in the chat box so that we will allow him, unmute him to share your views on the screen of Zoom app. Here is one announcement because it is not possible for us to be allowed everybody, but only just give the chats,
sir.
messages in the chats, so that we will exchange our ideas. And at the time of valedictory function, please give the feedbacks us, so that we can change what are the mistake in this national webinar we had done, can enhance or improve in ourselves. If anybody wants to be speak after this two resource person speak, then we will allow you to be exchange your ideas from the audience side also. Before that, you just give us a message on the chat box, so that we can admit your names. Within a fraction of second, the problem will be sorted out. Before that, I would like to say something. A good event never ends in the word. They take only the pause and keep us waiting for the next. I have no fear until the oxygen plant more trees available free. Plant a tree, oxygen is not a free. So plant some value. Plant in your new generation, they win a smell in the whole world as they grow up. This is the simple way to clean the environment, whether inside or outside. Everywhere there is a fragrance be spread. Dr. Naveen Congo sir, who is the professor and head department of microbiology, Dr. Hari Singh Gaur Vishwavidyalay, Sagar MP, will encourage or will enlighten in the subject of life science, microbiology. This is the need of tomorrow, because we are going such a pandemic period of a coronavirus, COVID-19. It is a need of our scientists to exchange ideas, so that some fruitful outcome will be there from it. There is a list of emails. We will share the PPTs. So many participants yesterday asked about the PPTs. I would like to share the participants' name over here. Dr. Ayushi, Dr. Amit Bhatt, Dr. Ananya, Mohan Kumar sir, Dr. Anupam, Pratap Singh sir, Dr. Ashok Patil sir is there in the attendee list, participant list. Dr. Avinash, Dr. Babita Rana, Dr. Chandrasekhar Pawan Patil sir is also here. Dr. Chirag Gujjar sir is also here. Dr. Damini, Dr. Damaji Vise, Dr. Deepak Fund. So many attendees are here. We apologize for only 2 seconds. We will start it.
Hello.
Yes,
KM
yes,
Bose
sir.
sir. KM Bose
Okay,
sir.
please. Yes, sir.
Kindly ask Parabia sir. He is ready. Then we will continue with Parabia sir.
Definitely, sir. There is a technical problem for the first resource person, Dr. Farjin Farabia, who is a second renowned person for the session. I would like to ask Dr. Farjin sir, are you there? Are you ready to deliver your lecture, sir, for Dr. Farjin sir?
Ready.
Dr. Farjin Parabia.
KM Bose sir. 1 minute sir. We got the PPT. 1 minute sir.
Okay, okay. I think so. Resource person sent their PPT here in our so very shortly the session will be start. Just hold a breath, because the without the interference there is no conference like that. No problem, sir. All the participants are very eager to listen the lecture, but due to the technical problems from their side, resource person side only, wait for 2 minutes. Very shortly we will start it.
So we
Here
got
there
the
is a
PPT. We
second
will
resource
start
person,
the
Dr. Farjit
session
Paradia,
in 2 minutes only.
who is from Farjit.
One minute, sir. KM sir, you continue.
Yes, sir.
Now I'm sharing the screen.
Yes, yes. You got the PPT?
Yes, yes. I share the screen.
Dr. Naveen Congo, sir. Yes, yes, yes. Now the PPT is visible from our side. Dr. Naveen Congo, sir,
Yeah.
are you hearing
Yeah.
us? We will operate the PPT from our side, sir. You just
Sure.
tell us
Sure.
next. You just start, sir, please. Dr. Naveen Congo, sir.
So once again, I regret the technical, flaw, and suddenly the computer also rebooted. So anyway, coming back to our discussion
Yes,
and
sir.
my talk for today, the title is like
Yes,
microbial
sir. Please carry on.
in generation of health promoting prebiotics, and what you can see below is like two waste products that come from agricultural practices. One is the sugarcane bagasse, the other one is rice straw, and then there are two molds. One is Aspergillus oryzae and the other one is Aspergillus quaternatus. So my talk is focused around how to valorize or maximize the use of those agro waste or the agro residues to develop value added products using microbial enzymes. I hope I am audible clearly.
Yes, sir.
Okay. So next, please. Next slide, please. Okay. So this is the picturesque eye view of my university that was on the screen. And this picture shows basically the concept of probiotic and prebiotics. So I want the young audience to pay attention to this slide and to understand what are these terminologies, what are these two terms which I am going to talk about. Like one is probiotic. So you can see the bacteria that are good for your health, and they thrive in your intestine, and then they promote your health by producing vitamins and so many good things. But then they are dependent upon these blue ribbons, as you can see, and these are selectively utilized by these bacteria for their growth. So you can see that these substances which are being selectively utilized by these probiotics are called as prebiotics. So coming back to the definition, probiotics are the good bacteria, mainly lactobacillus, bifidobacterium, and of course they include some molds also, like Saccharomyces boulardii. So the range is big, and then they grow there. They are like common cell. They live there, but they need some good promoting substances like prebiotics. So those blue ribbons shown in this picture are prebiotics, and these are the chemical substances. So ideally, how do you define prebiotics? These are the chemical substances that resist the digestion in the upper intestinal tract, and then they go up to your good bacteria, where the good bacteria selectively utilize them for their growth. By enriching the population of the good bacteria, that is probiotic, actually the bad bacteria go away. So I hope the concept of prebiotic and probiotics is clear. Now, what are basically prebiotics? Next, please. In terms of chemical structure and understanding. Next, please. Hello. Yeah. So again, the benefits of now the prebiotics as of today are limited to oligosaccharides. These are small segments of sugars. The sugars, 4, 5, 6 sugars, like fructo-oligosaccharides, mano-oligosaccharides, and xylo-oligosaccharides. So basically, as you can understand, that these are oligosaccharides that come from either fructose or xylose or mannose and so on. So you can have a look at their properties, and this talk regarding prebiotics especially become more relevant when we are in the time of COVID-19, and everybody's talking about boosting up their immune system and developing some antiviral properties. So there are always two ways of fighting with the disease. One is you kill the pathogen, which is very difficult in this case because viruses are obligate intracellular parasites, and as such, we don't have any antibiotic for this particular virus. Now, the other way around is what our other systems of pathies teach us, medicines teach us, like Ayurveda or say homeopathy, that teaches us to make our immune system strong by good diet and all that. So prebiotics come in between here. So they are not actually medicines; they are the supplements that can be taken up, and then you can see then your intestine is happy, and if your digestion is good, then you develop so many good properties, including a strong immune system. Next, please. Again, the benefits of fructo-oligosaccharides, which is one group of prebiotics with which we work, and basically these are like small oligomers of fructose. So you can note down these various. Of course, I have already shared with my PPT with the host, so you can always get those details. Next, please. Now, how do microbial enzymes play their role in oligosaccharide generation? So in today's this webinar, I'm very happy to share my experience sitting in my study room, and everybody is being comfortable. There has been no traffic, no rush, no formalities. So this kind of environment actually definitely can help us in real learning. So my point would be, especially with the youngsters, to take up the real good scientific things from this presentation which I have compiled for you people. So basically you can see that this slide shows generation of fructo-oligosaccharides using an enzyme named as fructosyl transferase, and the other one is generation of mano-oligosaccharides using a hydrolase that is endo-beta mannanase. So without getting into the jargon of enzymology, in the first case, what enzyme does is it actually adds up fructose residues to your normal sucrose. So you have one glucose and one fructose making sucrose, and then some again fructose comes with the help of FTase, another fructose come, and here we have FOS, that is called as fructo-oligosaccharide. In case of mano-oligosaccharides, actually this enzyme hydrolyzes the large mannan, which is found as a waste that I will be talking about in detail. So in today's PPT, in today's presentation, it's about enzyme production and characterization. Next, please. And then we go for finding out the oligosaccharides, and then we kind of find out their effect on the health promoting benefits. One moment, please. So basically these enzymes are being produced from the molds, and therefore I have used this title as mycogenic prebiotic synthesis. Next, please. And here comes a picture of the fungus, Aspergillus temeri. And you can see it's a common mold that we identified as Aspergillus temeri. And next, please. So enzymes involved in F1 generation, but I'm not going to talk about first two. We are going to talk about the third one, that is fructosyl transferase. Next, please. Okay, so here you can see in this schematic what I was talking about, how you make a fructooligosaccharide using fructosyl transferase. So what this enzyme does is it cuts the sucrose, the common table sugar, and at the same time it adds up the fructose to the end of the sucrose molecule. So you can see the generation of fructooligosaccharides as kestose, nistose, and fructofuranocil nistose. So basically, I would like you people to remember this particular slide, that these are the three most predominantly used fructooligosaccharides prebiotics. Next, please. And here you can see the over-the-counter available, combination of prebiotic and probiotic capsules that you can find. Even it is written in Hindi, pre and pro. So basically, one purpose of this presentation is to kind of make aware people who are many times using antibiotics. And then what happens when you're using antibiotics regularly, your stomach, your intestine gets devoid of the good bacteria because antibiotics kill indiscriminately. So when you are using some antibiotic, it is always advisable to use some prebiotic and probiotic. Together, these two make symbiotics. So I can what I wanted to show using this particular slide is that they are very much available commercially now, and they are quite costly as well. So with our intervention, with the intervention of microbiologists, we can lower down their cost as well. Next, please. So the objectives of this FTS work are like this, that we actually produce this enzyme in our lab. Then we go for their parametric optimization. I mean, we want to maximize the yield of the enzyme. And then we we check whether these prebiotics are actually promoting the growth of probiotics. That is the good bacteria. That I will come to you. Come in the next slides. Next, please. Okay. So this is the kind of ideation of this thought, that we select a fungus. We find that it is able to produce fructosyl transferase. You can see the dark red halo around this well in the very top left corner. And then we produce it in the flask. We produce the enzyme in the flask. And in this particular case, my student Ritumbara, she could find that the enzyme is stuck to the mycelia. So that was one revelation we came to know, and therefore we called it a mycelial fructosyl transferase. And actually, the enzyme is hooked to the mycelia. So basically, then we can also reuse it, as you can see in the last. So during this, then we characterize it by various means, and we analyze our products using various equipments that I will come to, and then we purify, and then we try to see how it affects the growth of probiotics. Next, please. So this is about the partial purification using ammonium sulfate fractionation. Next, please. Next, please. Okay. So let us not get into the numbers. I have already taken a lot of your time. I'm sorry about that. So basically, using ammonium sulfate fractionation, which is a very simple, cheap, and readily available technique, you can always purify enzyme to large extent. Next, please. And here is the picture that shows that the fructosyl transferase of this particular fungus, Aspergillus temeri, has been purified to 4 folds. And you can see that using a reagent called as TTC, you can always find out what is the molecular weight of your enzyme and how it is being, how many folds it is being purified. So in this PPT, in this slide, you can see the various, the various concentrations of ammonium sulfate being used, and eventually the enzyme getting fold purification, higher fold purification. The molecular weight of the enzyme was about 75 kilodaltons. And this has already been published in a journal called as Food Chemistry in 2019. I will be very happy to provide the reprints and everything. Next, please. Okay, then it comes about how to characterize the product. So basically, to make young audience more, to make them understand it more clearly, you can see a flask. The flask contains sucrose with little bit of minerals. And then what we do, I call fungus as a good bioresource, and a bioresource which is present everywhere, which is omnipresent. Even in Dhulia or in any part of Maharashtra, you can isolate beautiful molds. And then these molds can, when you put them in right milieu, in right place, they're going to produce wonders for you. So in this particular example, you can see that fungus, we inoculate this fungus in this flask containing the table sugar at high concentration, of course, say 20%. And then we allow it to, kind of shake. And then after some time, we see that the fungus is able to produce. If you are able to see this HPLC, you are able to see that the fungus transforms the sucrose through fructosyl transferase into fructooligosaccharides, which I told you, which are like prebiotics and which are a costly commodity, a costly supplement. Next, please. Okay, then we have to get into the different kind of instrumentation to prove our point. Then this particular, the green one shows that how you can characterize the fructose or fructooligosaccharides using NMR, and the one which is showing small, mountain-like shape. These are the FTIR diagrams of the fructooligosaccharides, FTIR analysis. So this particular slide shows how you can use NMR and FTIR for quantifying and kind of analyzing your fructooligosaccharides. Next, please. Okay, like I said in the beginning, because the enzyme was a mycelial fructosyl transferase, it was already attached to the mycelia. So the student had an idea that why cannot we use it as an immobilized enzyme, and it was correct. So you can see that the mycelial FTase was being used, recycled for at least 10 number of times and producing different amounts of nistose and kistose, which are fructooligosaccharides. Next, please. Next, please. Yeah. So these are again some graphs showing how we optimize. These are the numbers. I think we can move on. Next, please. Again, next, please. Next. Okay. So one interesting thing, that how do we see that these oligosaccharides are actually promoting the growth of, the good bacteria? So then we have to perform analysis again. And in this case, we inoculate 2 lactobacilli, which are good bacteria, in the fructooligosaccharides produced by our mold, produced by our enzyme. And then we can see the growth at, say, OD 600 of the lactobacillus being enhanced. And one thing, and the second thing that we analyze is that the lactobacillus, when utilizes this fructooligosaccharides, produces good amount of short chain fatty acids, which are again good for our health. The abbreviation for short chain fatty acids is SCFA. So basically this graph shows that how you can promote the growth of lactobacilli, one, and second, how you can see that the lactobacilli itself is producing a lot of short chain fatty acids using this fructooligosaccharides. Next, please. So again, some analysis using NMR where that says that actually you can see the short chain fatty acids using a very big instrument called as NMR. Next, please. Okay. This is again some interesting study that shows that you can use the biogenic, silver nanoparticles and immobilize your enzyme onto that, and then again reuse that enzyme for the production of fructooligosaccharides. Next, please. Then you again use various techniques for characterizing, just to kind of inform the young audience that once you have prepared a nanoparticle with a enzyme that is hooked to, nanoparticles with enzyme attached to it, then you can analyze it using scanning electron microscopy, atomic force microscopy, and of course you can check the presence of the elements using EDX. Next, please. So basically, once you have an enzyme nicely immobilized onto your nanoparticles, you can again reuse it, just like we use the mycelial fructosyl transferase. One thing I wish to point out about the enzyme is that it's very kind of tedious and time-taking, and of course expensive to produce an enzyme. So basically, once you have produced it, all the enzymologists want to kind of reuse it, and immobilization is one such technique that allows reuse of the costly and otherwise not available enzymes. Next, please. Next, please. Okay, so these were the key findings of this study, which I already discussed. This part was more about, fructooligosaccharides. Next, please. And this is our publication that has come in food chemistry last year. You can always send me an email and get this paper from for your, if you are interested. Next, please. So very quickly, I would be getting into the other part of my study that talks about mannooligosaccharides. So in the first part, we talked about fructooligosaccharides. These jargons may bore you, but kindly bear with me, because I have been working with these, say, around say 6 or 8 years. So please take due interest in this and try to understand what I'm trying to discuss. Especially, I'm requesting the young audience. And the idea is that how the microbial enzymes, especially the fungal enzymes, can be used in various ways to deliver, important and quality commodities. So this title says how we can exploit the mannan. So next is, what is mannan? Next, please. Okay, I'm sure most of you have seen at least some of these, and without taking much time, basically mannans are gums. So, and one very common gum is guar gum. What you can see in the first, in the right, in the left corner, the green, beans, guar gums. Jala guar fali mantoban. And then you can explore. And then secondly, one part is this, and the next is that the oil producing industries like palm oil producing industry and copra oil producing industry produce a lot of palm kernel cake and a lot of copra meal that contains a lot of mannan. So next, please. This is basically the idea of mannan. Obviously, any good student can understand that the name mannan signifies the number of mannose, that it is made of mannose. Mannose is again a hexose, and you can see a variety of mannans shown in this particular slide: linear mannan, glucomannan, galactomannan, and galactoglucomannan. So there is a huge range, and it ranges from a pure homopolymer to a heteropolymer. Next, please. Okay. Now, when the nature creates a polysaccharide, it also creates, the enzymes. So basically, the mannan is storage and structural polysaccharides in some plants. And then we have these endobeta mannanases that hydrolyze the backbone of this, long polymer made of mannose to produce various, sugar moieties. Next, please. So in our case, what we are trying to do, we want to produce mannooligosaccharides from mannan. So like I said in the beginning, what are these oligosaccharides and why we are interested in them? Because we want to use oligosaccharides as prebiotics. Next, please. Okay, so this work we started in 2015 when Hemant Soni helped me in isolating this number of molds. So I am very happy to share this slide, and you can see a number of molds: Fusarium, Solani, Cladosporium, and Aspergillus, and then there are some thermophiles, and then there are some new ones. And at the end of the names, you can see National Fungal Culture Collection of India, which is situated in Agarkar Research Institute, Pune. So we do isolate number of molds and we do deposit them to this culture collection, and that is how we make them safe, and we can always retrieve it whenever we require them. So basically we screen molds for different enzymes. In this particular case, you can see there are some numbers that say this fungus produces this much amount of the enzyme. Next, please. So when it comes to actually looking at that, how you can say that what are the products of your hydrolysis. So you can use an instrument called as HPLC, and in this particular slide you can see that the figure A is the standard that shows M1, M2, M3, and M4. Like in case of fructooligosaccharides, we had nistose, kistose, and all that. In this case, we have manotriose, manotetrose, and so on. I hope I'm able to make some point here. So you can see when you hydrolyze mannan with different, enzymes, then you get different products. Next, please. Okay. So then there is a trick which we call as optimization. Like every student wishes to optimize his or her performance by studying, by optimizing their answers, their writing skills, their vocabulary, their understanding of the subject. So similarly, when you put a microbial resource like fungus or a bacteria in a flask, you need to provide a good environment. You need to give it a good kind of support so that it listens to you. It kind of helps you in producing large amount of the products in which you are interested in. So in this particular case, like I said, the waste product coming from the copra industry, that is copra meal. A lot of southbound states produce a lot of copra meal, which is a waste. So we have used Aspergillus styrus oryzae and other Aspergillus mold for the production of mannanesses. Next, please. Okay, so here is one picture that shows that this fungus is Aspergillus quadrilineatus, and this fungus was isolated by Rahul Suryavanshi, and then he went on to identify it using 18S. Now everybody knows, most of you must be knowing, that the identification nowadays is based on molecular taxonomy. So this dendrogram shows how related this particular fungus isolated in Sagar, Madhya Pradesh, is like Aspergillus quadrilineatus. And then it is very nicely able to colonize copra meal in solid state fermentation. Again, a very cheap technique that can be used in any college or any science department. Next, please. So here are some umbrellas showing some figures that show that how you can optimize, like I said, the production of the enzyme using this technique called as response surface methodology. So most of my students are able to learn it where they use pH and moisture in case of solid state to optimize the production of enzyme. Next, please. Then these are again some numbers that say that once you optimize, you can actually enhance the yield. Say in case of Aspergillus quadrilineatus, up to 10 times, almost 9.9 times, or 5 times, or 4 times. So this is the effect of optimization in case of microbial cultures. Next, please. Again, some HPLCs showing products of the enzyme action. Next. Next, please. So likewise, you can see You can always go on hydrolyzing various gums. In this particular slide, you can see the names of the gums: lactose bean galactomannan, guar gum, that comes from the guar falli, right? Konjac gum, palm kernel cake, and copra meal. These last two are the agricultural, agro-industry waste coming from the copra and palm oil. And the other ones are the plant, mannans. Next, please. All right. Now, again, like I said, a good approach for an enzymologist is to save his enzyme for reuse. So what we do is we immobilize our enzymes on various substrates. In this case, particularly, what is this AOP? This AOP is aluminum oxide pellet. So basically, you can buy aluminum oxide pellet from any good company, and then you can hook your enzyme using glutaraldehyde, and then you can characterize these things. Like the first pictures are same, the other one is like XRD, the other one is like FTIR, and then you can get into the multi-scale characterization of your enzyme hooked to some particular matrix. Why? Because then you are able to reuse it. As you can see, if you are able to see the small balls in these Eppendorf tubes, these are the balls that we use. So the idea behind immobilization, like I already said, is like reuse. Next. Next, please. Yeah, again, these are showing some characteristics of the immobilized enzyme. So, right. So once we are able to immobilize the enzyme, then you can see that you can go on producing, your monooligosaccharides again and again in this, the chart that is showing the bar graphs. And here you can see at least you can recycle the enzyme 10 times, and you can see that it produces M3, that is mannotriose and mannotetraose, many times. Next, please. I always wanted that some of the students must have interrupted me and asked me something, but of course I'm not able to look at the chat. Yeah, let's come back to the presentation. And well, this particular slide shows a very interesting, aspect. All the students know that the polyacrylamide gel electrophoresis is used for the protein separation. But in this case, Uttam Jana, that this paper is already published in a good journal, Bioresource Technology, Jana et al. In 2018. And you can see that the sugars, the oligosaccharides, being fractionated or being separated using polyacrylamide gel electrophoresis. So we were very happy to develop this, not for the first time, but to use this technique in our lab for characterization of the end products of mannan hydrolysis. And the technique is called as fluorescence assisted carbohydrate electrophoresis, that is FACE. So you can see very beautifully resolved M1, that is mannose, M2, that is mannobiose, M3, that is mannotriose, and then M4 mannotriose. Like I said, the higher DP oligosaccharides are being used for, are being used as prebiotic oligosaccharides. Next, please. So this is now I'm coming to end of my PPT, which I put up for the young students especially. Now, how to prove that these prebiotic mono-oligosaccharides are actually imparting the health promoting benefits. So this was the next idea. This was the next agenda of our work. And then we took up various exercises, and I will show you in the coming slides. Next, please. Right. So the good properties of the mono-oligosaccharides. So what we do is we hydrolyze the mannan and then check it for scavenging activity, for the antioxidant activity, for the chelating activity, for the anti-cancer activity, for the growth promoting activity of the probiotic bacteria, and so on. Those are the first in this particular slide. You can see the antioxidant properties of the mono-oligosaccharides. Next, please. Okay, then this is a very important aspect of oligosaccharides. Prebiotics are known for, I mean, they are being explored for the anti-cancer activity, especially, anti-cancer activity that can be imparted in the intestine, because these are like, they will travel to the large intestine. So in this particular slide, we have shown that the CACO2 cell lines, the colorectal cancer cell lines, are being affected by the mannooligosaccharides adversely. So the cells die out to large extent, and we use 2 concentrations of mannan hydrolysates, and you can see the role of mannan hydrolysates in lowering down the number of cancerous cells. Next, please. So again, very fondly, I'm happy to share the common sources of mannan, and you must have seen aloe vera, you must have seen guar gum, and it is also found in coffee. It's also found in methi. हमारे यहाँ कई बार कहा जाता है कि मेथी बहुत अच्छा है, एलोवेरा बहुत अच्छा है. क्यों अच्छा है? क्योंकि उसमें कोई ऐसा ingredient है. So mannan is that possibly one of those ingredients. Next, please. Next. Again, how we hydrolyze mannan. Next, please. Again, multi-scale characterization. Next. I won't go much into details of these. Next, please. This FTI. Okay, we have also used this NMR for characterizing the hydrolysate of the various mannan coming from various mannans after mannanase activity. Next, please. So again, the numbers showing that how you can identify mannotetrose using NMR. Next, please. Again, this slide shows the hydrolysis produced, the mannooligosaccharides produced using aspergillus coordinatus, bringing down the number of CACO2 cells, that is showing the anti-cancer activity. So you can see 2 concentration of mannooligosaccharides changing, bringing the number of cancer cells down. Next, please. Okay, now this is very interesting, and a very simple experiment that can always be performed in a college also with limited facilities. You can, the wells are basically, they come from, they are like ELISA well plate, and the bar shows the kind of biofilm being produced by the lactobacillus. So we have used 2 lactobacilli, Lactobacillus delbrueckii, Lactobacillus acidophilus. And you can see very well in this picture, which was made by Rahul Suryavanshi, again, one of my PhD students. And that shows that now the copramyl hydrolysate in the bar, which is going high and showing the highest, film promoting activity. And in this case, in case of B, it is LBG, that is locust bean gum. So basically these 2 hydrolysates, when they are, put to use, when they are given to Lactobacillus delbrueckii and Lactobacillus acidophilus, they form better films. Now, these are 2 good probiotic bacteria. So if they are able to grow happily, if we are able to enrich their population using mannooligosaccharides, then possibly your mannooligosaccharides can be used as prebiotics. We already published this information in food chemistry, which will be coming up very soon. Next, please. Okay, so one way of, I'm coming to the fag end of my talk, and I'm very happy, and I regret that there was a delay due to some technical snag, but eventually I hope that I am able to make some point with the young audience. What I'm trying to say now is that how these lactobacillus and other probiotics help in promoting good health is by producing some short chain fatty acids like acetic acid, propionic acid, and butyric acid. So this particular TLC shows generation of these short chain fatty acids by these 2 probiotic bacteria. Next, please. Okay, so once you have these short chain fatty acids, the intestine is happy, and if your digestion is good, everything is good. Next, please. Again, these hydrolysates help in generation of more number of amino acids through lactobacilli. So as evident in case of, next please. All right, as evident in this case, okay. So here we come to the conclusion that we can produce mannolisaccharides using endo-beta mannanases, and sourced from various fungal sources. In this particular presentation, I have showed the endo-beta mannanases coming from Aspergillus quadrilineatus and Aspergillus oryzae, and then we can also see how they are able to promote good health by promoting the film formation ability of the lactobacillus, by enhancing the amount of short chain fatty acids, etc. Next please. Okay, now coming to the end, these are some of the papers coming from our lab. Immobilization of Aspergillus mannanase, published in Food Chemistry, and the next one is again coming in 2020, where we have evaluated various mannolisaccharides. The last part of this talk is coming in this paper, the 2020, very soon. Next, please. Okay, so some of the old work from 16, where we used Aspergillus terreus and other things to hydrolyze the mannans, published in 3 Biotech and other journals. Next, please. Okay, then this is where I got interested. The title at the bottom, you can see characterization of hemicellulases. So basically, mannanases are hemicellulases, and from this, where we picked up this field, and we're trying to do something good in that. Next, please. Okay, screening part showing number of moles and their mannanases in zymogram, published in Engineering and Life Sciences. Next. Okay, this is Uttam Kumar Jana's paper from Aspergillus oryzae, again published in Bioresource Technology. I have discussed this phase technique and zymography and all that. Next, please. And then some characterization of these mannolisaccharides with the help of my physicist friend, Dr. Day, and published in Chemical Physics Letters and some NMR journals. Next, please. Next, please. Okay, so this is the work regarding Aspergillus tuberculosis. This is another work where this fungus is being used for the bioethanol. I have not talked about this today, but I wanted to show you this, that we also do this kind of work where we produce glucose and then we ferment it to bioethanol. Next, please. And this work has already been discussed. Aspergillus quadrilineatus mannosaccharide generation. Next, please. Okay, then this is a Malbranchia cinnamomia, which is a thermophilic mold. Again, specifically generating mannotetraos. Next. And the earlier part where we discussed generation of fructooligosaccharides using Aspergillus temeri was again published in Food Chemistry. And I have shown you most of these diagrams, including the analysis, immobilization, and the zymography. Next, please. Okay, next, please. So some more Mannan work which has just come up in International Journal of Biological Macromolecules in 2020. Mannolisaccharides derived from agro-waste mannans. Next, please. Okay, these are my, I can say, colleagues or my scholars. So with which I work and from whom I learned. So they have been helping me in learning so many tricks, so many techniques, and getting so many good molds, beautiful molds. So I invite all the participants to my university. Whenever there is a suitable time, you can always come and explore the possibility of discussing some good science with us. Next, please. Thank you for a very patient hearing. I regret the technical snag, and it was a pleasure talking to all you. And I will be very happy to answer the questions right now, or maybe if there is a dearth of time, then you can always email to me. Thank you. Namaskar.
Thank you, sir. Thank you very much. Very informative lecture regarding the role of nanoenzymes, fungal enzyme, microorganism. Now, Dr. Naveen Congo sir has explained the role of microbial enzymes in the generation of health promoting prebiotics. In this, Dr. Congo explained to us the usefulness of a gut-friendly bacteria like lactobacillus, beneficial effects of oligosaccharide generated by the action of gut bacterial enzymes were also explained. Finally, he explained the process of valorization of mannan rich agro waste for MOS generation, which has many applications like anti-cancer activity. So he said that FOS and MOS are only two prebiotics class found in the pet intestine, that is a fructo-oligosaccharides, where the fructose as a main sugar chain and monogo oligosaccharides. Meanwhile, in the chat box, we are getting lots of messages. Dr. Anita Mudavkar ma'am chats valuable information given by Dr. Naveen Congo sir in this pandemic period of COVID-19. Now the session is open for the question answer. If anybody having the question to be asked directly on the screen, please raise your hand so that we can unmute you. Really, it was a nice lecture in the chat box. We are listening or we are reading that. So many chats. Any question from your audience side? Please.
Sir.
Yes, sir.
I request Manisha ma'am to ask question.
Dr. Manisha madam.
Okay, I think she is not available now.
Yes.
Second is Sheikh ma'am.
Make them unmute and their video should be visible in front of the audience. Dr. Sheikh madam, please ask your question. If it is not possible, you can ask by chat box also.
Yes.
The session is open for 2 minutes.
Okay, sir, continue. I think she is not available.
1 minute. There is question?
Sir.
Okay, sir. No problem. Thank you very much, sir. Thank you very for the informative presentation in front of our audience. We will see you also in the future also, sir. Thank you very much. It's a nice presentation. Now we will move further for the next presentation. Now I call upon Dr. Sambhaji Patil to introduce today's second resource person, Dr. Farjin Parabia, sir, Associate Professor, Department of Bioscience, Veer Narmada South Gujarat University, Gujarat. So Dr. Sambhaji Patil, Department of Botany, SSUP Science College. Please introduce Dr. Farjin Parabia, sir. Please, sir.
Okay, thank you, sir. Namaste and good afternoon to all. Welcome to this session of webinar. Here I am very glad to introduce the speaker for the session, Honorable Dr. Farjin Parabia. Dr. Farjin Parabia is currently working as Assistant Professor of Plant Biotechnology from Ashok and Rita Patel Institute of Integrated Study and Research in Biotechnology and Allied Sciences, India. He did his PhD in botany from Sardar Patel University, Vallabh Vidyanagar, Gujarat in the year of 2008. He also served as visiting faculty in Ayurvedic College, New Vallabh Vidyanagar and B.V. Patel Pharmaceutical Education and Research Development Center, Ahmedabad. His area of research instead focuses on agricultural biotechnology, plant molecular biology, plant tissue culture, anti-malarial drug testing on in vitro culture of Plasmodium falciparum, medicinal plants with taxonomic identification, reverse pharmacology and ethnopharmacology, and phytochemistry, ethnobotany and pharmacognosis. Dr. Farjin has published 5 books, 16 research articles in journals, and 15 presentations in national and international events. He also invited as a resource person in number of scientific meetings He successfully supervised one doctoral thesis, three MPhil theses, and 28 master's theses. He also attended the number of seminar, workshop, and symposium. He is a number of he is a member of an editorial board in World Research Journal of Biotechnology and Space. He is a life member of Medicinal and Aromatic Plant Association of India. So I am again welcoming him on this platform. Thank you.
Thank you, sir. Thank you, Dr. Sambhaji Patil. Now, I directly call Dr. Farjin Parabia, sir. Before that, I request Principal Dr. M.V. Patil, sir, to welcome Honorable Dr. Farjin Parabia, sir, by offering a bouquet. Thank you, sir. Thank you very much. Now, I request Dr. Farjin Parabia, sir, please deliver your valuable lecture in front of our audience. Dr. Farjin Parabia, who is Associate Professor, Department of Bioscience, Veer, Narmada, South Gujarat University. Dr. Farjin Parabia, sir.
Hello, good afternoon.
Good afternoon, sir.
All of
Please
you.
start.
I am audible.
Yes, sir.
So let's share my screen. Able to see my slide?
Yes, sir.
Okay. So actually, right now I'm working as an associate professor in Department of Biosciences. Actually, previously I was there in Ashok and Rita Patel Biotechnology. So right now, my lab and my students, we are working on the evaluation of known phytochemicals through molecular docking to predict possible interaction with novel coronavirus, which is right now known as a COVID-19. So coronavirus it is actually a big family of a virus which has a RNA as a genetic material, and it can infect a broad range of vertebrate, which include even birds and other mammals also. And not only respiratory problem, but it can generate another disease also, like that. Anthracis is caused by coronavirus in pigs and cows. Right now, it is spreading in human, damaging alveolar of a lung, and death is happened. And it is released and pandemic endemic occurred in December 2019. So it is known as a coronavirus disease 19 COVID-19. Actually, this is a new virus, but most of its sequences are matched with the SARS virus, which was occurred in 2002. So because of that, it is also referred as a SARS CoV-2 coronavirus 2. And right now, none of the targeted therapy or vaccine is available for this particular virus. It is a big problem. So before entering into the docking, first we have to understand what are the receptors and virus structural and non-structural proteins are associated with it. So you can see this particular slide is showing that how this particular virus is spreading. You see the virus enter into the human cell through the receptor which is known as ACE2 receptor. It will bind with that receptor, release a single stranded RNA strain into the body. From that particular RNA, transcription and translation happen. Another virus particles generate, and gradually they infect our entire respiratory system, and death also occurs. So I am mainly focusing on a structure in which the spike protein, you can see this is a spike protein which is a outer membrane structure of a virus. It is made up of S1 and S2, two subunit. This is an enlarged view of this particular protein. This is a protein through which it come into the interaction with our cell receptor, which is known as the ACE2 receptor. This is a angiotensin converting enzyme 2 receptor. So if we can disturb this binding to any kind of herbal therapy or even some kind of allopathic drug also, then virus entry can be inhibited. The another genes, RNA, having only a single strand of RNA, length is also only a 30 kb of a length. And the major genes present, which are the protease, replicase, helicase, endoribonuclease, and a major spike structural protein. So we can design a drug or select a phytochemical from a database who can block either a receptor or either any of these protein. It can be served as a therapeutic agent for this virus. So there are the two line of therapies present. One is the vaccine development, or second in which you block any of the accessory protein with related with SARS-CoV-2, in which the host receptor proteins are there. There are two host receptor proteins. One is ACE2 and second one is TMPRSS2. Another set of a protein, they are the virus structural and functional protein. We can inhibit that also. Vaccine development is targeted by S subunit of that code protein. It has a two subunit, S1 and S2. S1 subunit, it will bind with a receptor, and S2 subunit, it will bind between the host and cell receptor membrane. So as I have said that host cell, there are two receptors. You can see this particular paper published by Hoffman. SARS coronavirus 2 cell entry depends on ACE2 and TMPRSS2, and it's blocking by a clinical proving protease inhibitor. So actually this TMPRSS2 is a protease enzyme, and if we can block that enzyme, then also the virus entry is not possible. So we can search for a phytochemical who can block that entry. So let's understand these two receptors. First receptor is a angiotensin converting enzyme 2. Angiotensin is a substrate for this enzyme. From that, this enzyme have a name. So if we block this enzyme with anti-ACE2 antibody, then also the infection is prohibited. And if we can search a phyto compound which can block this receptor, then also the virus entry can be stopped. This is a structure of ACE2 receptor. Now both the receptor has their own role. TMPRSS2, this receptor, it has a activity as a protease enzyme, and it has a name also, transmembrane protease serine 2. So these particular protease activity, it will be first act on a S protein or of a viral membrane, and this particular protein has been cleaved. So this is called as a S protein priming. When this protein cleaved, then only it can bind with a membrane, and then the virus entry is possible. So Huffman has blocked that particular entry with use of a serine protease inhibitor and another compound which is named as a chemostate mesylate. And this compound is right now under the clinical trial also. These are the number of genes which is present in a virus. There is a one website, Zhang Lab. They have constructed a three-dimensional structure for only COVID-19 genes. You can see these are the numbers of genes present. I am taking a selected certain genes and proteins on which right now we are working, and majority of the research group are working on that protein. The first protein, it is known as a main protease protein. This is a major protein, and docking is started by this protein. This is an inhibitor N13. It is a proven inhibitor of this particular protein. This is responsible for a replication and transcription process. So if this protein is blocked, the virus purification is blocked. The another compound name is ibislan. This is also having a proven activity to block this particular protein. Another important protein, it is a SARS coronavirus and SP12 polymerase protein. This is a basically polymerase enzyme, and it at a time of working, it bind with the two factors. One is a NSP7 and another one is a NSP8. So this protein is called as a non-structural protein. From that, it get the name NSP. So this is also very, very essential for replication and transcription process. This is a prefusion 2019 and coronavirus spike glycoprotein. This is a S protein, and this spike protein is present on a membrane of coronavirus, and this is a protein which attach with ACE2 receptor and make the entry into the human cell. And the affinity of this particular protein is much more higher than the previous SARS coronavirus, and that's why this virus is highly infectious. This is a RNA-directed RNA polymerase enzyme. This also play a major role in replication and in transcription process. This is a helicase enzyme. This will do the DNA and RNA duplex unwinding process. So, who supports the scientifically proven traditional medicine? So, right now, virus do not have any kind of medication. What we treat by some kind of a symptoms. Instead of that, if we search a certain plant phytochemicals, we perform the docking. And if we find out some kind of a positive result, we may propose government for going some kind of a clinical trial. We have already did a same kind of a work. That was a one DST sponsored multi-institution project in which we have developed one anti-malaria medicine from Calotropis procera. There were two formulations we have developed. One named as a Arcmil, second one is sold as a Calomel. There was a multi-institution project. Professor I.L. Kothari was my guide. He was a PI from Sardapati University. And Professor Parabia, he was a PI from Veer Narmad South Gujarat University. And it was a collaborative project between this was our team at a time of that research. So right now, even anti-malaria medicines, it is a one of the candidate to fight with this COVID. You can read this paper. Chloroquine is a potential inhibitor of SARS coronavirus infection and spread, in which they have tested on a virus cell line, and they have find out that ACE2 receptor glycosylation process is affected by chloroquine, and that they have proved in a Vero E6 cell line. Same thing with Artemisia annua. They have observed that alcoholic extract of this plant, it was served as a potential herbal medicine for SARS coronavirus. So these are the certain examples of plant we are using from many years in Ayurveda to cure malaria, like Sarsha, Chiraita, Andrographis panicula, which are known as a Karyatu. Hemidactyon excelsum, that is a brahmachal, that is a tree from which the bark is utilized on malaria. Artemisia maritima, this is a species of Artemisia which is occurred in Gujarat. So if we want to do this kind of a research, first we have to have a list of a plant, then we search for a phytochemicals from that. Then we derive the 3D structure of those phytochemicals, and then we select a certain virus related protein, and then we are going for a molecular docking. So this is a number of plant right now. My one of the student Rachna is working on this. Adatoda vesica we know as a Urdusi, Tinospora cordifera, very good immunomodulator garo plant. Terminalia bellerica beda, very good on a bronchial cough, and a Zingiber officina is well known. Pipal longum lady pepper, that is also utilized to cure cough and cold. Curcuma longa, very popular one. Calotropis procera and Gigecia already having proven antiviral activity, also anti-malaria. Piper nigrum, that is a black pepper utilized in lots of Ayurvedic preparation. Piper beetle, that is our Indian plant, and also utilized for cough and cold. Datura mickel, Solanum xanthocarpum, Kantakari. From that, Kankari aule is there in the market. Cardiophylleta, Basilicum, Oscillum centellum, Nigeria sativa, Vidania somnifera, Alstonia, Sporadis, Anicostema axillaria, It is known as Mamejo. From that, the Mamejo Ghanvati is available in the market. This is a plant also utilized as a anti-diabetic plant also, and also on a fever also as a directa indica. So right now we are working on these particular plants. So first we have to search the phytochemicals. So there are lots of databases are available, like Dr. Duke database, Nepralet database, then Indian Medicinal Plant Phytochemistry and Therapeutic database. You can search out the phytochemical which is available from any of these references. You can search the manual research paper also. Such kind of manual database are available and published in this particular review article. This is specially written on that. OpenSS metabolomics database. You can search such kind of a database from this paper. Then we have to derive the 3D structure of those phytochemicals. So those 3D structures are also available on PubChem, DrugBank, ChemSpider, and Campbell database. Certain compound whose 3D structures are not available, we have to construct them by utilizing certain software like Marvin Sketch, ChemMax, BioVina, PubChem Online, Argus Lab. So these are the facilities through which we can construct the 3D structure. Now we have to select a viral protein and download it 3D structure that is available on RCSB database, Swiss model, ZangLab, Uniprob, and CBI. There are lots of other sites for which we can download that structure. These are the software available for molecular docking: Visual Studio, Autodoc, Samsung, Pyrex. You can utilize any of this service. If you want to read a review for those software for molecular docking, such reviews are also available. This is one of the example of online database from which we can search a compound. Suppose like this, we give Aadha Thoda Vesica, and it will give a list of compound. Now from that particular list, we can search for its three-dimensional structure from PubChem. If the structure is not available, through the software, we can construct that structure. We download that 3D structure. Now using another service, we convert into the PDB format. PDB format, this is utilized for a molecular docking. So this is one online open basal software through which we can convert our SDF file of secondary metabolite into the PDB software. Same way, we download the protein 3D structure also from the database, and then we utilize docking software. This particular example is regarding to the Samsung Connect software. We have opened that particular protein into that software. After that, you remove water molecules from that. You load your phytochemical and then perform the docking. It will show you where it will match and give you that particular binding energy. So right now, these kind of research, lots of lab is working on that. Like within on, they have find out that it will be disturb the virus interaction with our receptor protein. Same way, curcumin, it will block with main spike glycoprotein, which is present into the fourth virus. Another group, they have screened 22 phytochemicals and find out 6 having affinity with a spike glycoprotein, and another 2 has affinity with a main protease enzyme. From Mauritian medicinal plant, they have screened 67 molecules, and they have find that small, which having a good affinity to bind with the main protease enzyme. My other colleagues are also working on the same. They have docked, and whatever the in silico docking results are that they have combined with our university website. And any person want to search the docking result, they can utilize that service also. So we have GSBTM sponsored Param Shevak, which is a supercomputer, and with the help of that, we can perform such kind of work. So thank you very much for a patient hearing, and now I am hand over to the organizers.
KM Borse, sir. KM Borse, sir.
Thank you, sir. Dr. Farjin Paradia has given a very fruitful and delighted knowledge that regarding the enzyme and phytochemicals. Dr. Farjin Paribia sir explained about the evolution of known phytochemicals through molecular docking to product predict possible interaction with the novel coronavirus. It is the need of today's. Now we shall proceed further with the question answer session. The audience are welcome to put forward their queries, if any, before our esteemed speaker. We will unmute the mic of audience whose hand is raised for question answer session one by one. So if any question from audience side, please ask. Even though you can ask question in the chat session also, chat box also. Very informative lecture by Dr. Farjin Parabia sir. From audience side, if any question, please ask. There is option of raise hand near the share participant box. More, click it and then click on the raise hand so that we can unmute and give the spotlight on the participants. If anybody wants to ask a question to Dr. Farjin Parabia sir, please raise your hands. I think so there are lots of chats which are coming over here. Good informative lecture. Thank you very much. Valuable guidance. They wants to ask us, is it possible to share the PPT?
Yeah, sure, definitely.
Yes sir, yes sir. We will share this PPT to all the participants.
Sure.
Oh. Okay, thank you very much sir, Dr. Farjin Parabia sir,
Thank you.
for giving your valuable guidance and time to us.
Thank you.
There is one question. So before that sir, I would like to ask in the chat box, there is one question, which is a more effective and surface protein receptor blockage or vaccine regarding the coronavirus treatment?
Right now, many people are working on that. For molecular docking purpose, we can utilize all these proteins to dock and see the result.
Yes. Okay. Thank you very much sir.
Thank you very much.
Now,
Chambers, sir.
okay sir, thank you.
So there is one question from Swapnil Joshi.
Swapnil Joshi wants to be asked.
Yeah.
Please give spotlight to Swapnil Joshi sir.
Swapnil sir, you can ask your question.
Yes.
Hello.
Sir, one question from Dr. Swapnil sir.
Yes, please.
Good evening, sir.
Good evening.
Yeah, sir, I just want to ask you, what are the possibilities of developing vaccines in the near future?
Vaccine protocol itself are taking 18 months, and many company has claimed that they have developed and it is under the clinical trial. But we are not sure up till what it is reached into the Indian market and reach up to our doorsteps. So right now the precaution is better than a cure, but the protocol itself is taking 18 months.
Okay. Okay, sir. Thank you.
Okay. Most welcome.
Yeah.
What sir? Continue.
Yes, sir. Thank you very much, sir. A very good question asked by Dr. Joshi. Really, it's a good and informative presentation in front of us. Everywhere there is a life science on the surface of this earth. We can see as just like the carbon. In an enzyme, there is a carbon. In the drugs, there is a carbon. In the fats, there is a carbon. In the protein, there is a carbon. In the food daily which I eat, there is a carbon. In the hair, there is a carbon. In the land, there is a carbon. In the sand, there is a carbon. Everywhere there is a carbon. We can say that kan mein hai carbon. So very good and informative lecture delivered by Dr. Farjin Paribia. Thank you very much, sir. Thank you.
Thank
We will very soon see you once again in the future also. Thank you, sir.
Thank you.
Honorable delegates who blessed us with their presence, took out our valuable time of their busy schedule. I must mention our deep sense of appreciation for Dr. Farjin Parabia, sir, Dr. Naveen Congo, sir, Dr. Minu Parabia, sir, Dr. Falguni Shet, ma'am. And I also heartily congratulate all the participants for their active participation in this event. Now I call upon Dr. Praveen Mahale, sir, Department of Zoology, SSB Science College, for the vote of thanks.
Thank you, sir. I, Dr. Praveen Mahale, on behalf of SSVPS Samstha's late Karmavi Dr. P.R. Gogre, Science College Dhule, and entire organizing committee head. I must mention our deep sense of appreciation for Honorable Babasaheb Sunalji Patil, MLA Maharashtra State and Chairman SSUP Samstha Dhule for providing all the necessary facilities during the organization of this webinar. Further, we are grateful to Honorable Professor Naveen Congo for delivering a very informative talk on knowing COVID-19, a big challenge. Thank you, sir. Thank you for your very informative lecture. And we are also grateful to Dr. Parabia for today's talk on evaluation of known phytochemicals through molecular docking to predict possible interaction with novel corona. I express my gratitude to our principal, Honorable Balasaheb M.V. Patil, sir, for providing encouragement and all the necessary facilities during the organization of webinar. I also express sincere thanks to our National Advisory Committee members. Those are Professor A.B. Chaudhary, sir, Dr. D.S. Pokre, Professor Pipe Khare, Dr. S.N. Nandan, Professor D.A. Patil, Professor V.L. Maheshwari, sir, Professor N.K. Patel, sir, Professor P.R. Puranik, sir, Dr. R.J. Sayed, sir, Dr. B.D. Bose, sir, Professor D.R. D.A. Kumaut, sir, Professor P.S. Doa, sir, for giving valuable suggestions during the organization of this webinar. I also express my gratitude towards our conveners, Dr. Sandhya S. Patil, Head Department of Botany, Dr. D.L. Fund, Head Department of Zoology, Dr. Harshal Bhambre, Department of Biotech, and all organizing committee members who are taking efforts to organize this such a very wonderful event, and Dr. Rakesh Devre, Dr. Arsur sir, who are taking efforts to organizing and all the such event. I also express the sincere thanks to us entire teaching and non-teaching staff of our SSUP Science College. I also extend my thanks to all the participants who joining us by various applications like Zoom, YouTube, and Facebook Live. I also for their enormous cooperation during this session. Thank you very much. Now I request to all participants to stand in respect to nation for the national anthem.
Thank you, Dr. Praveen Mahale. Now, I would like to request all the audience, please, everybody stand up for the national anthem. Thank you very much. We will share the feedback form link on the WhatsApp group, your email address. Please fill up feedback form as soon as possible so that it is easy to us to share, to give the certificates to all the participants and those who already registered themselves. We will provide the link on the WhatsApp group. And in future, definitely we will again organize same kind of webinars. If any there is mistake from our side, from our technical sides, please forgive us. Definitely we will enhance in future. Thank you very much.